Hello, my name is Killian. I am a second-year graduate student in Dr. Tracy Grew's Cognitive and Functional Wellness Lab. This is my Discover 2023 entry entitled Affective Cognitive Control in Adolescents with Epilepsy and the Impact of Comorbid Depression. These analyses are made possible by our collaborator, Dr. Crystal Cooper at Cook Children's Medical Center. Epilepsy is one of the most common neurological disorders, while depression is one of the most common psychiatric disorders. Approximately 50% of individuals with epilepsy have depression when compared to 10 to 20% of the general population. Each of these disorders can significantly disrupt affective cognition, which are cognitive processes involving emotional stimuli. As noted in the graphic, there are substantial overlaps in brain regions involved in affective cognitive processes and regions implicated in epilepsy and depression. Despite these observations, there has been little investigation of affective cognition in adolescents with epilepsy and comorbid depression. The study examines affective cognitive control involving successful inhibition of one response in favor of another in the presence of conflicting affective information. We had two goals for this exploratory analysis: evaluate differences in affective cognitive control between epilepsy, EPI, and typically developing TD adolescents, and assess relationships between task performance and depressive symptom severity. We hypothesized that adolescents with epilepsy would have longer reaction times and poorer accuracy than typically developing adolescents, and task performance would be significantly associated with level of depressive symptom severity. The current study is a secondary analysis of individuals with epilepsy and typically developing adolescents recruited from Cook Children's Medical Center. Our study included 8 EPI and 16 TD adolescents who were matched 2-to-1 to epilepsy participants based on age and biological sex. Ages range from 10 to 19 years old. TD adolescents varied from epilepsy participants by no more than 3 years. The emotional Stroop task was used to assess affective cognitive control. This task involves presentation of either happy or fearful faces, with the word happy or fear displayed across the face in a congruent or incongruent conflicting manner. Participants are asked to respond to the face. Both accuracy and reaction time are measured. The Patient Health Questionnaire 9-item, or PHQ-9, was used to assess self-reported depression severity. This is the demographic and clinical data table. Matching allowed for an equal number of males and females in each group. The mean age was 15 and the mean PHQ-9 score was 7. Accuracy and reaction times for emotional reactivity to conflict were calculated by subtracting congruent from incongruent trial performance for overall and emotion-specific measures. A series of independent samples t-tests were used to compare accuracy and reaction time measures between the two groups, and Pearson R correlations were conducted between performance scores and depression severity. The top 3 figures are different scores in accuracy for incongruent minus congruent trials. Accuracy did not significantly differ between the groups. Those with epilepsy had greater variance on accuracy for incongruent trials of fearful faces, which was significant. The bottom 3 figures are different scores and reaction times for incongruent minus congruent trials. In the left graph, we see the reaction times for overall conflict indicate that the EPI group has slower reaction times than the TD group when processing emotional conflict. Group differences in the conflict scores for happy faces were also significant. The EPI group took longer to resolve conflicting stimuli for happy faces than the TD group. These findings suggest that both groups could perform the task well, shown by their equivalent overall accuracy. Those with epilepsy may have impaired emotion processing for fearful faces, shown by their reduced accuracy but similar reaction times. The epilepsy group was also slower to process emotional conflict for happy faces, suggesting an impairment in emotional conflict reactivity. This is the first study to investigate emotion conflict control in adolescents with epilepsy compared to a typically developing cohort. Despite the lack of significant findings in the correlational analyses, our population had epilepsy subjects with moderate to severe depression that are in line with expected rates. Sample variance violated Levine's test. Results should be seen as exploratory and interpreted with caution. In addition, we did not correct for multiple comparisons. The sample was small, so there may have been insufficient depression severity range to allow for meaningful correlations. These data demonstrate greater variability amongst adolescents with epilepsy on affective conflict performance. Additional testing with adequate sample size is needed to further examine differences. The comparison of affective cognitive control versus non-affective cognitive control in epilepsy is needed to better clarify the role of cognitive control deficits in epilepsy during adolescence. Understanding the impact of epilepsy and depression on affective cognitive control domains may guide development of interventions to improve affective cognition and related outcomes in adolescents with these disorders.